Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Metabolon, I'd like to welcome you to the Evolution and Future of Metabolomics Technology, Surprising Discoveries and What This Means for the Future. My name is Elizabeth Lamb, and I'll be the host and the moderator for today's event. I'd like to introduce our presenter for today. He is Dr. Mike Milburn, PhD, Chief Scientific Officer of Metabolon. Welcome, Mike. The presenter ball is yours.
So, good morning, everyone, and thank you for joining us today. Just as a quick introduction to my background, I'm the Chief Science Officer at Metabolon. I've been with the company for approximately 13 years, and I'm responsible for the R&D, technology development, lab operations, both clinical and our research studies. So, I'd like to just start off by kind of giving you an overview of what the presentation is today. So when I was asked to put together this webinar, I thought what I might do is kind of give a perspective of the last 13 years at the company and how we've developed our technology a little bit, and then more about, some of the surprises and some of the technology areas, discovery areas that have come to be important in the company. And then I thought I'd look a little bit to the future and see where, we think a number of these developments are going to lead and be important and compelling in the next 3 to 5 years, let's say. So that's the focus of this presentation. I did want to just mention real quickly that we have a number of webinars that are available online through our website, or if you just Google Metabolon and YouTube, that are more focused on our technology, precision metabolomics, different applications. And I encourage you to look at those if you have a deeper interest in anything that we're doing. So the first part of my talk is really just going to be 4 or 5 slides, kind of giving you a little background to the company and what we do. Just as a little background, we're based in the Research Triangle Park here in North Carolina. We're founded in 2000. The lab operation started around 2002, 2003. So we've essentially been really focused on developing a technology that can profile and generally profile all of the small molecules less than 1,000 Daltons, in any kind of biological sample. So that's actually been the mission of the company from the very beginning. We're now on our kind of 4th version, if you will, of our platform. It's a very advanced technology now. It's very industrialized, very automated, although we're still automating, and I'm going to talk about that today as well. We did acquire a company in Germany last year to facilitate more of our European studies. You might have seen a press release this week, actually, of our platform that we've put in China. So we have some of these satellite locations also in Qatar for being able to perform our analysis in different countries. And of course, our largest lab operations is here in RTP, where we have approximately 200 employees. And I've already kind of talked about the mission of the company. What I wanted to talk about a little bit was just why we got into metabolomics. Back some 13, 14 years ago, a lot of companies, a lot of technologies were developing around genetics and genomics, transcriptomics, and really trying to better understand DNA and what the genetic sequence and mutations mean. We really felt at that time that if you could develop a technology that can more phenotype a biological system, so characterize its metabolism, how it's using energy, really trying to get a picture of what's currently happening in the state of the organism, that would be very important in the future in better understanding these DNA mutations and how we're all different genetically, but also it would have interesting applications outside of just working with DNA information. And that's certainly been the case. We've found a tremendous number of applications, and that's going to be the first thing I'm going to talk about, and kind of the surprising discoveries we've made is just how applicable this technology is for a lot of different areas. And then, of course, we also get a lot of information about the environment, xenobiotics, drug metabolites, and things like that from how we run our technology as well. So when you combine these different types of information, whether you're looking at a cell or you're looking at a blood sample or a tissue sample, you're really getting a good overview of metabolically, phenotypically, what's happening in that in that organism or in that cell. So that's really kind of why we got into this, and I think it's really bared a lot of fruit now in terms of how important this technology is, particularly with understanding underlying genetics. And I'll spend quite a bit of time talking about that in a minute. So just a brief slide about how our technology works. And as I said before, this is kind of our fourth version of the platform, which is pretty advanced in terms of the coverage of biochemicals that we can perform now, as well as the speed and accuracy of measuring these molecules. So essentially, we work with groups, and we do approximately 800 studies a year with different clients and academics, government labs, and commercial pharmaceutical companies, biotech companies, and others. And we work with those groups on how to develop a study, a proper study design, and then essentially those samples come into the company. And once the samples come here, we do all the processing, all the data analysis, and even the biochemical interpretation. So every sample that comes in is extracted for the biochemicals in that sample, whether it's tissue, cell culture, blood, urine. We've worked with over 200 different types of matrices for performing this kind of analysis. Once we extract that sample, then there's essentially 4 aliquots that are made of that sample, and then each aliquot is run on 4 different chromatography mass spec-based methods. And the reason why we run those 4 different methods is simply that there's no one good chromatography mass spec method that can measure all the different physical properties of small molecules. So by measuring these 4, we get a good coverage of all the very polar to lipid to nonpolar molecules. What we've done and what we've spent a lot of effort on is really developing the software, the libraries, the informatics behind being able to take that essentially raw ion information, and each of these methods is generating between 30,000 and 40,000 unique ionic pieces of information. And then we essentially call those down into the individual biochemicals that we tentatively identify in each of these methods. And then we've developed software that generally can go in and look for and QC which platform, which method is going to measure that small molecule, and then perform the final data set QC, so that at the end of the day, in 100 microliters of blood today, we typically measure around 1,000 biochemicals, both in terms of identifying them and then quantifying them for the groups that we work with. And then we can take that even further and then perform a biochemical interpretation or a biochemical data analysis of that data and report back not just the raw data, but also the statistics, the biostatistics, and the biochemical interpretation of that data. Because we provide so much of that information and we perform so many of these studies, we've published, or our data has been a part of well over 1,000 peer-reviewed publications, and you can access those again through our website, and they're broken down by different types of articles. Articles we published in cancer, articles on our core technology, and those are all available as well through our website. So that's essentially how the entire analysis works. It takes us approximately 6 to 10 weeks to turn around a data set. So we can process upwards of 800 samples a day now. So the studies go very, very quickly, even studies as large as 1,000 to 10,000, 20,000 samples. We perform relatively routinely and process approximately about 130,000 samples a year now. So it's a pretty big operation, and we've been kind of working at this and industrializing this for the past 15 years. So, and just real quick on this slide, I'm not going to spend a whole lot of time on this slide, but it really does speak to how rapidly and how advanced the technology has become. So if you look at the bottom right panel, that's the metabolite coverage. So that's essentially the molecules that we would routinely measure in a couple hundred microliters of blood running our different methods that are available on those samples. So even some 5 years ago, we were measuring upwards of 500 to 600 molecules, and today we can measure anywhere from 1,500 to 2,000, depending on the study in a blood sample. And when you look at a wall chart of biochemistry, we're actually covering 80% to 90% of all the biochemicals on those wall charts. So it's biochemistry is relatively finite. So there's a unique set of biochemicals that our human body can synthesize. Then there's the metabolites that come from bacterial metabolism, and then there's also xenobiotics. And when you look at when you have a technology that can look at all those different classes of molecules simultaneously, we're now getting a pretty complete picture from where we were even 6 or 7 years ago. And the precision that we're doing with doing that analysis with is much greater as well. So on the bottom left are the RSDs, if you will, relative standard deviations of all the small molecules that we measure in technical replicates that we run on every study. Every 7 sample is a pooled technical replicate of those samples that we're looking at. And we can measure then the relative standard deviation of each molecule that we're measuring, from total extraction to quantitation, with those technical replicates. And because we do that and we run those standards as well as other QC standards in every study we run, that's really allowed us to continuously improve and develop the technology for the past 15 years or so. And on that top bar where all the blue dots are, this is essentially all of the different pieces of unique software or informatics or new hardware detection devices that we've added over the last years of developing the technology. And again, if you're interested in more detail about the core technology, how we've built our libraries, how the informatics works, the QC curation process, those are again available on other webinars that you can get through our website. So, and I've talked a little bit about this, but this is our biochemistry wall chart that we've developed, and now we're getting a pretty complete coverage, pretty complete picture of all this biochemistry, and as well as with the precision and the breadth of what we're measuring today, we can actually look at clinical samples now and look at an individual and how they deviate from an average normal population. So that's something that's very new. I'm going to talk about that, and I think it's a big future of the technology development and clinical applications of metabolomics, as well as diagnostic applications of metabolomics. Most of what I wanted to talk about in the next 20 or so slides is really kind of what we've learned about different applications that have been, somewhat surprising or just even more impactful than we originally thought, and then a little bit about where we think the future in some of these areas are going to go. And the first one I wanted to kind of highlight was just the different application areas that we worked with in different companies and different types of companies that we work with, many of which we never really anticipated having important metabolomic applications. So we always thought that this would be important technology and research and pharmaceutical research, biotech research. But, some of these areas have been really surprising. Just to highlight a few of these, one of the areas that perhaps wasn't as immediately obvious to us was the work we do in consumer goods. So this includes cosmetic companies, food companies, and things like that. So we do a tremendous amount of work looking at skin samples and the type of samples that we didn't anticipate looking at, really helping companies figuring out whether their products are improving health, improving lipid metabolism, decreasing inflammation, things like that. And that's a big area for us that we didn't necessarily conceive of 10 years ago or so. Another area very much like that is approximately 15% of the studies we run today are in bioprocessing and developing different expression systems for different cell types. So we do a lot of work in that area looking at the raw materials that go into these bioprocessing additions, which can be quite expensive once they get scaled up. We help companies, figure out which cell lines might be most metabolically stable for developing further in terms of a product manufacturing standpoint. We've done a lot of studies looking at limiting ingredients, if you will, in the media and things that get depleted over time, that if you replenish those things, you can get improved yields or sometimes improved protein product. So, that really is an area that we never really anticipated being as important or as big an area that we would end up working in, and it certainly has been a big surprise for us. And then I think the two other areas I just want to highlight, because I'm going to talk about these in more detail in the next few slides, is the phenotyping and genomics. So approximately 50% of all the samples we run today are really using our data to better understand which mutations in the DNA, in the genetics, are penetrant and biologically relevant. And so we do that not just for clinical samples, but we also do that with large retrospective studies looking at the genetics of individuals and better understanding which mutations are important, and in some cases, which mutations may have initially been thought of as being biologically penetrant, but from our phenotypic data, we don't see as strong a phenotype. And so I'm going to spend a bit of time talking about that area, because that's really been something that's exploded, particularly in the last 4 or 5 years. And then the second area is the microbiome. So one of the things that I think we've helped characterize over the last 5 years is how important and how many biochemicals are, how much of your metabolism is controlled by your bacteria, and how often these bacterial metabolites are turning up to be not just important biomarkers, but perhaps even mechanistically some of the signaling mechanisms that are going on with different disease conditions and with different nutrition effects and with different diet effects. And so I'm going to spend some time talking about that area, since we've been doing a lot more work in that space than we really anticipated some 10 years ago. So this is really just kind of the application areas, and there's lots of application areas that we work in, but I just wanted to highlight a few that have been kind of more surprising in terms of the degree of work that we do in that space. One of the first things I want to talk about is this whole how you combine genetics and genomics with metabolomics, and why that's turning out to be so important. And I wanted to start off by just giving a little background to what I'm talking about. So some 100 years ago, this paper was published in 1909, which was really the first by Archibald Girard, which was at Barts College, which was really the first paper that highlighted this link between having an aberrant biochemical, in this case an amino acid, and having that linked back to having a genetic effect. And of course, this is long before Watson and Crick and understanding the DNA structure. This was just knowing that there was a genetics underpinning to having these strong biochemical disease effects, and that you could actually screen for these genetic effects by looking at biochemical measurements. And what he proposed in this paper over 100 years ago was that these are just the gross anomalies of a genetic disease, but that we all have mutations or underlying genetics that give rise to unique biochemical individuality. And that a lot of the individuality or a lot of the biochemical levels that we maintain homeostatically in tissue and in blood are actually coming from our underlying genetics. Now, I think some 6 or 7 years ago, when we first got into this, a lot of folks thought of this space as being perhaps not very tractable with metabolomics. And because I was, very much involved with this when we were first doing these initial studies, the idea was that biochemicals or metabolism is too changing or too willy-nilly with time of day and diet, that you would be able to pick up on these genetic mutations with individual biochemical measurements. So to really answer that question, we collaborated with 2 groups. One group was at King's College, Tim Spector's twins cohort, where he's been collecting samples and doing DNA genotyping as well as genome sequencing on these twin populations, and being able to get access to those samples and basically ask the question, can we look at those cohorts, those studies, and find alleles that are relatively common alleles that are, sufficiently abundant in these populations that associate or statistically correlate, if you will, to different biochemical levels in the blood. And so this was a study that was published in Nature. We also have another publication in Nature Genetics, which was really the first kind of large-scale study asking the question, how penetrant is your underlying genetics? So we looked at the twins cohort, and we also looked at a cohort in Germany, which was the Cora cohort with Karsten Suri at the Helmholtz Institute. And so the idea was we have these two independent populations, one's British, one's German, and we have all this genotyping data for each one of these, and can we look at which alleles in the German cohort are associated with these different biochemical levels and how that compares to the twins cohort. So what you're looking at on the top in this graph, on the top here is what's called a Manhattan plot. So these are all the chromosomes, and then you're looking at each of these red dots then is where there's a highly associated biochemical to an individual allele. And so in this paper, we not only looked for statistically significant associations between a biochemical level to that allele in the German population, but the only ones we described also had to be matched in this UK population. And there were 32 that were significant in both of these populations and were the same allelic variation. And so that went on to this Nature Genetics paper, which we published about 4 or 5 years ago, now looking at a cohort of about 8,000 individuals. And this is now 132 associations in both of those cohorts. So some of the interesting findings of this was that most of these genetic associations we're finding actually turned out to make perfect sense. So when you look at the levels of proline in these populations, the biggest contributor to what your level of proline is actually what allele you have for proline dehydrogenase. And the same thing's true for glutamine, glutaminase, and a number of these other biochemicals and genes. And as I said, when you looked at the entire list, some 90% of them actually made perfect sense. But this was really entirely unanticipated, because what it really was saying was that, in fact, your DNA or your genetics is highly penetrant to what biochemical levels you actually have, and that they can, whether it's, these are all fasting samples, but even, how they were collected and measuring these fasted samples on these individuals, we could still identify these genetic variations in these biochemical levels. So this was, really the start of something that's turned out to be really important for a lot of studies we run. And this is a really interesting slide, which I don't have time to go into, but it's basically showing that, these biochemical changes are actually titratable. So this is the German cohort for one particular biochemical, and you can see the difference in the concentration of that biochemical, whether you're heterozygote or homozygote for a particular mutation. And that was true in the twins cohort as well as the German cohort. And it was true for multiple biochemicals, really suggesting how important your underlying genetics is to, your biochemistry being able to be measured and phenotyped in these blood samples. So we've gone on, and if you go to our website, there's some 20 publications now looking at combining genetic information with metabolomics, as well as doing that now on clinical. So looking at individuals that have been sequenced and looking at their metabolomics. This is another paper published much more recently with human longevity, where we looked at the same twins cohort, but now we had 3 independent measurements over 20 years of each of these twins. And we did whole genome sequencing in this case. So I don't have time to talk about this entire paper. It is published now. And some of the findings, this is a finding that's showing that a lot of these genetics for these twins were actually very hereditable. So a lot of the genetic variation we saw between the genes to the biochemicals were from hereditary, not from environment. This is something you can only calculate looking at twins. But one of the things I did want to point out was some very interesting findings with a few of these twins. So if you look at the levels, this is dimethylglycine. And we just simply asked the question here, if you look at some biochemicals that had aberrant levels or very high levels for some of the twins, could you explain that genetically, or was there a genetic linkage to why that biochemical was different? And so this was a set of twins that, again, there were 3 measurements, 3 different blood draws of these twins over 20 years. And it's the same set of twins have these highest levels of dimethylglycine. And when you look back at these 2 pairs of twins, you actually found they had a double mutation. So they had a loss of a stop codon and a missense mutation. And that's what's giving rise to them having this high level. And what's kind of fascinating by this is that the next highest sets of twins also had mutations. And interestingly, though, these are heterozygous, so these are carriers for this mutation. If you're homozygous for this particular mutation in this gene, it's actually a pretty debilitating inborn errors of metabolism. So they're likely somewhat phenotypic with having these strong effects. But again, these are heterozygous effects that are very penetrant and very biochemically measurable across these populations. So that's just one example that I won't go into this example, but this is showing that we found other mutations, that there are canonical mutations that give rise to certain aberrant biochemicals. We found these same aberrant biochemicals, but there were different mutations. And when you look at the crystal structure of this particular enzyme, you can actually see these mutations were in the same region as the canonical mutation. Just to summarize that one piece, what we've generally found is that this unique genetic information and combining that with metabolism can give you a lot of information about an individual or these populations, not just how penetrant a particular allele is, but also whether that mutation is actually biochemically relevant. Some of these mutations get reported back to patients because there's been a finding previously, but we sometimes don't see the biochemical evidence that phenotype is important. And then in other cases, there are other genetic gene variances that are actually quite important that get missed because they're just not as well understood from the purely genetic side. And as I said before, this is now more than 50% of all the studies we run is combining or samples, I should say, not studies, is combining this genetics with metabolomics. And there's a lot of information, a lot of other papers that have been published in this space, and I encourage you to look at our website and our bibliography to find some of these. So moving on, that was a lot of the data that we've done in genetics. I think the future in that space is really that we're now talking to groups about mapping 100,000 or even upwards of 1 million genomes to metabolomics. And I think what that's really going to do is allow a very detailed mapping, if you will, of genetic mutation to biochemical phenotypic changes. And these phenotypic changes, these biochemicals that we're seeing linked back to DNA, it's really a full spectrum of amino acids, sugars, lipids, but also xenobiotics and bacterial metabolites. And so I think there's going to be a very rich data set generated over time of uniquely mapping a lot of individual genetic mutations to these phenotypic outcomes. Just a little bit about the microbiome. Of course, there's a lot now in the publications, peer-reviewed publications, also in the general news about the importance of your microbiome. I think from our perspective, one of the big findings has really been the last 3 or 4 years that there are just so many biochemicals that we measure in blood, upwards of 200 biochemicals we routinely measure in every blood sample that's just coming from bacteria or just coming from your gut metabolism. So we often get the question, how do we know that? How do we know these are just coming from bacterial metabolism? Well, there's really 3 answers to that. One is these biochemicals, there's not an enzyme in humans that can synthesize them, but there are bacterial enzymes that can synthesize them. The second point is that we've done a lot of studies looking at antibiotics treatments of animals, antibiotic treatments of humans, and these, the vast majority of these within a very short amount of time will essentially go to 0. They're not measurable. So we also know from that kind of thing. And then we've also looked at animal studies where animals have been grown up in a clean environment. And again, these molecules are missing. So there's a lot of evidence from our perspective that these are purely bacterial metabolites. that's interesting, but they're circulating at fairly high levels in blood, and we're finding more and more that these are being important phenotypic changes in disease and diet effects and nutrition effects. And so again, I think this has been a surprising area of how much metabolomics can contribute to this space. And one of the points that I often make is that you can have very diverse genetic populations of bacteria, but they can give rise to the same, metabolic homeostasis. But that dysbiosis of the microbiome may not be as apparent from just the genetics standpoint. But we can clearly see when these molecules get aberrant levels from what is considered to be normal. In many cases, that's due to some other type of effect like chronic fatigue or some other disease or health consequence. And so I think this is going to be a big area of the future, just monitoring these molecules and better understanding why they're contributing to health and disease. And just, some examples of that. This was a really nice paper that was published in Nature in 2016 from the Weizmann Institute that was using our data. And they were doing a lot of other analysis, but essentially what they were looking at, if you look on this bottom left, was taking mice on normal chow and taking mice on a high-fat diet and showing these weight differences. But then they showed that you can actually put these mice on a high-fat diet followed by normal chow and then followed by high-fat, in some cases a normal chow, and you get what's called this yo-yo effect where, if just by inducing these high-fat diet conditions, these mice, when they go back even on normal chow, they still have substantial weight gain. And this has been commonly called in the literature the yo-yo effect of dieting. So there's a persistence here from being on high fat and gaining sudden weight, that when you try to go back to a normal diet or a lower calorie diet, you're actually still having this significant weight gain that's persisting much longer than you would expect from the time period that you were on the high-fat diet. And so they looked at a number of effects in here that were really quite compelling. They looked at the microbiome, the genetics of the microbiome. I don't have time to go into those different aspects, but one of the things they did look at was the metabolomics. And this, I think, was one of the key pieces of that paper, where what they showed was that during these high-fat diet where these mice became obese, there were some molecules that were at pretty high levels in the feces. These were 2 flavonoids that were at very high, not very high levels, but at pretty significant levels in the feces. But that once they went on these high-fat diets, and then even after 15 weeks of being off those high-fat diets, they had persistently lower levels of these 2 flavonoids. And there's a lot of literature about flavonoids, the positive health benefits of many of these flavonoids. But it was interesting that these 2 particular flavonoids had this persistence of, even after 15 weeks of being off the high-fat diet, being significantly lower levels. And I think to me, the really interesting aspect of this paper was that they went back and actually added those flavonoids to the normal chow diet. And so when you look at the addition of those flavonoids, you actually then you have a positive effect on ameliorating that yo-yo effect. So these animals go back to normal weight, and that's compared here in the green and the yellow. If you give the flavonoid, in the absence of the flavonoid, you still have this persistent, significant higher weight gain. And of course, they did a lot of nice controls in this paper in terms of food intake and other things like this. And so it's a really interesting finding that not only is these 2 molecules potentially a biomarker of this yo-yo dieting effect, but that these bacterial metabolisms, if you will, of these diet compounds might also be mechanistically linked to having this ability to gain weight, even maintain weight gain even after short periods of high-fat diet. So I think that's a really interesting microbiome example where we looked at how the metabolomics, or how they looked at how the metabolomics could contribute to the understanding of that and finding these biomarkers. Another study I just wanted to point to that I thought was very interesting was a paper published in Cell in 2013, where we looked at an autistic mouse model. And the short answer to that model was when you looked at the disease model, the autistic model, there was actually one metabolite that was 40-fold higher levels in the disease model versus controls, and that's 4-ethylphenol sulfate. And that molecule can only be bacterially synthesized from tyrosine. And they showed that not only was this potentially a very interesting marker of that effect, but if you actually gave this molecule to normal animals, you could actually induce some of these autistic behaviors. And I think, that's really a key concept that whereas we're learning more and more about how important the microbiome is to health and disease, we always have to keep in mind that it's not as if the bacteria is actually, going to these different locations and having these effects on the brain, but it's how they're influencing metabolism, how they're changing biochemistry. And, this becomes a very exciting area in the future, is better linking, again, the bacterial genomics to this phenotypic metabolism information. One of the things I did want to mention, though, in terms of the microbiome is that this is a big space in the future, I think, for novel metabolism. So a lot of the metabolites that we're uncovering that we know that we've measured, for instance, in 5 or 6 years, and now we're identifying what those molecules are, many of these are turning out to be other bacterial metabolites. And so, what we put this slide in showing that I think in the future, there's going to be a lot more of combining this metabolomics to the genetics of the microbiome. But I think this is a huge discovery area as well in terms of new biochemistry that, is probably very important to different health consequences. And we're just kind of scratching the surface biochemically understanding this right now. And I call this the natural product discovery area for the microbiome. So finally, just to kind of wrap up real quickly on the last two areas, I mentioned earlier about using our technology clinically. And so what I mean by that is in the last couple of years now, we've developed the platform technology where we can take an individual blood sample and measure all the biochemicals in that blood sample and then relate that back to a normal population. So we can give for upwards of 1,000 biochemicals, we can say which of your biochemicals or which of your biochemistry pathways are altered relative to a normal population. And if you look at some of our press releases, in fact, even last week, we had a press release of using this clinical metabolomics for helping to diagnose difficult disease conditions that have gone undiagnosed. We call it Meta-UDX, which is helping to diagnose or analytically giving information about how potentially diagnosing difficult to understand disease conditions. And the concept here is that, typically when physicians are measuring diagnostic assays today, they're really looking at a relatively small set of clinical panels. So even if you think about your physical, you might have a metabolic panel, a lipid panel, but there's literally upwards of 1,000, 2,000 molecules that you can measure additionally. And that's really what we're talking about, is kind of shining this lamp, if you will, on a larger blanket of biochemicals and then trying to understand how much additional information or how much more detail can we give about health consequences or even diagnosing diseases by looking at these larger groups of molecules. And so this is just an example of a paper we published with Baylor, and they've been one of our big collaborators in developing this space. We have a number of publications with them, particularly on childhood genetic disease. But this is an example of looking at a wall chart for an individual child. So this was a previously diagnosed child in a paper we published as a control paper where they had maple syrup urine disease. And you can see how when you look at all the biochemicals that are aberrant, so if it's decreased, it's blue. If it's increased, it's red. And the size of the circles, the number of standard deviations away from the average of the normal population. You can see how that upper left quadrant lights up. And that's essentially leucine, isoleucine, and valine metabolism, branched-chain amino acid metabolism. And the particular pathway or the particular enzyme that's disrupted is the dehydrogenase that's involved in maple syrup urine disease, genetic disorder. So we can generate these kind of maps for any individual. We've used this now to help develop or identify new genetic diseases as well in some of these pediatric populations, and we're now using these in adults as well. And I think in the future, and you can also score these. So typically, many of these things are being measured in relatively small numbers. So you might just measure an amino acid or one other metabolite. We're measuring all the metabolites in these pathways, or many of these metabolites. So you can come up with these algorithmic scores that are much more specific and potentially higher signals for uncovering these diseases. And I think this kind of area is going to be much more important in the future, where you're starting to think about using clinical metabolomics and the applications of clinical metabolomics. And the challenges for us, and I think the challenge in the future for the field, is going to be how to turn around these data sets to better, to do it in a time frame that allows for patient management. How to interpret these data faster, so that a physician doesn't necessarily have to spend a week trying to understand biochemistry to give the appropriate information back to a patient, or how to move forward with that patient's treatment. These are all, this is all very new right now, but I think, in the future, this is going to become a really big area for metabolomics and how we develop this clinically. Finally, I just wanted to mention that a lot of biomarkers have come out of this technology in the last 10 years or so. These are diagnostic tests that we've developed. There's underlying unique biochemical biomarkers that have been a part of these. And so we often get asked the question, what have you discovered new biomarkers? And the answer is yes. And there's lots of underlying publications, particularly in the Quantus IR IgT test and the development of that, and how we identified the biomarkers discovered. These last few slides is just showing this work that we're doing in this kind of clinical space using genetic information on individuals and combining that with phenotypic information. And we're doing that work. I mentioned the publication with Human Longevity, and we're also working with Arabelle and Padia and others in this space. I talked about some of the tests that so this is our MetIMD test. So this is a test that we can run on genetics of children to use this technology to help diagnose genetic disorders, as well as we've been doing this with adults now and looking at the underlying genetics of adults. And that's what the MetaUDX, the undiagnosed disease test that we put out a press release last week on. So again, I think that's going to be a big area for us. Some of you may have seen this ad that was out that was really trying to make the point, it's a play on Match.com, but really making the point of how important I think the future is with combining genetic information with metabolomics to better understand these, not just genetic diseases, but what these mutations mean from a health consequence. So finally, on this last slide, you've got some information about our next webinar on May 24th. And if you have any questions after this seminar, and of course, or this webinar, and if you want to refer anybody to them, it's all available on our website, or Metabolon, just searching on Metabolon YouTube. So I just want to thank you for your time, and happy to take any questions.
Thank you very much, Dr. Milburn. Yes, we have quite a few questions and not a lot of time, so we'll get started. Our first question is from Ryan. Are these platforms standardized for whole blood analysis, or typically limited to plasma or serum?
We can use the whole blood, plasma, or serum. They're pretty interchangeable. We can also use dried blood cards. So all of those are available, and we have a lot of experience with all those different sample types.
Our next question is from Marika. What is unique about Metabolon's approach versus other players in the field of metabolomics?
I think a lot of the metabolomics that's done that I see published in the literature is still looking a lot at the ion feature data. So I talked about that we take a sample and we run it on 4 different methods and sometimes 5 different methods. And what we're generating is 30 to 40,000 unique pieces of ionic information. And I think a lot of the groups out there still work with the ion information and do statistics on the ion information, and it's very difficult to do that. A lot of the molecules that we measure may have 40 or 50 redundant pieces. In other words, the ionic information 40 or 50 pieces of that ionic information is coming from one molecule. So there's a lot of redundancy in those individuals. There's kind of an over-concentration of certain molecules in that analysis. And in many cases, we have groups that come to us that say, I found this really interesting ion feature data, but I'm still trying to figure out what the molecule is, and can you help us with that? And so, one of the unique aspects of our technology, where we've invested a ton of money and a ton of time and really developed a lot of informatics, is we don't work with the ion feature data. We've developed these, libraries of upwards of 5,000 standard chemicals run at multiple concentrations on all 5 of these methods with the informatics and the QC software, so that within minutes, literally, of running a sample, we're converting all that ion information to individual biochemicals. So, if you're interested in more detail about that, there's a very good presentation that was given by one of our scientists here 2 months ago that's available as a webinar that I referred to earlier, that's called Precision Metabolomics, and it really highlights how our technology is kind of unique in this space relative to most groups doing metabolomics.
All right, the questions are pouring in now. So our next question, you mentioned finding new microbiome metabolites. Could you elaborate a bit on how this is done?
I probably didn't explain that really well, but so part of our library are molecules that we've measured over the last, 10 years or so, but we've still not identified what they are. So they have all the characteristics of what a chemical standard is, but we haven't necessarily identified what that molecule. So 13 years ago, when we started doing this, the vast majority of the molecules were these unknown molecules. Today, it's relatively small. It's probably, less than 20%, in many cases, 10% of the molecules we're measuring are in this space. And we elucidate about 50 to 100 molecules a month. So and many of these we're having to synthesize because these not chemical standards. So one of the things I tried to point out in the talk is that a lot of the molecules today that we're identifying, that we've been measuring over time, are coming from bacterial metabolites. So there's not much, there's really very limited information chemically that they really exist metabolically, but also there's no chemical standards. And so that's really what I meant by, I think, a lot of the future of this microbiome space is not in just how important, measuring these molecules are going to be in understanding the bacterial populations, but also that this is probably a right area for covering new biochemistry that exists.
Our next question is, besides a fasting sample, do you recommend any other restrictions to account for the changes in metabolites over time?
Yeah, probably the best answer to that question is the most important thing is that the samples are handled reproducibly. And so if you're doing fasting, or you're doing non-fasting, or you're collecting EDTA plasma, or you're collecting heparin, just making sure that the sample type and how you're collecting the samples is as consistent over time. And we do a lot of studies that are non-fasted samples, and we can get quite good data as long as the biological variation, is high enough, and it often is. But there's really, the biggest recommendation would really be, handling samples and consistently collecting the samples across the entire spectrum of the study. And if you're interested, we have documentation on sample preparation, how much sample we need, how to collect sample, where to ship it, all that kind of thing, and that's also available on our website, or you can email that email below.
All right, our next question. Do you have plans to follow some of the heterozygous cases revealed by WGS longitudinally?
Yes, I think this is a big area in genetics, is understanding these heterozygous mutations. If you think about it, we have probably so many mutations that, fall underneath this, whatever you want to call it, heterozygous carrier status, that typically, because you're a carrier, you think, well, I'm fine, there's no phenotype to it. But I think in many cases there likely is. And there's actually very good literature out there that's already, so if you look at sickle cell disease, if you're heterozygous for sickle cell disease, that does not mean that you don't get sickling. And in fact, one of the most common unfortunate deaths that occur in high school football players is due to sickling from heterozygote individuals. So these carrier status or these dominant mutations from a heterozygous standpoint are likely going to be more phenotypic, particularly as you age or have other disease effects. And I think this is going to be a really important area in the future, particularly for metabolomics.
All right. Is this technology being used to also map diseases without a clear genetic connection, that is, complex disease?
Again, this undiagnosed disease area that we're working in, I mean, many of these individuals are people that have, for instance, chronic fatigue. They've gone in and out of different clinical testing. Some of them have done quite extensive clinical testing. Some have done genetics. That's just one example. Some of these are inflammatory conditions where, there's probably some underlying condition that they just are some underlying effect, and they've just gone undiagnosed over many years. And so it's very early days for that. I'm not trying to suggest that we're figuring out every case every time we run a sample, but I do think that this is going to be a really important application of the technology, is using it clinically in this. And again, the hurdles or the limitations right now is going to be about turnaround time. It's going to be around how to use it for patient management. And I think the most important thing is how to more automate the interpretation of this data for these physicians to use it.
All right. I see we're coming up here on the hour very quickly. We do have a lot more questions, so what I will do is plan on forwarding those along to you, Dr. Milburn, so that you can answer them via email. I'd like to thank Dr. Mike Milburn and the folks at Metabolon for sponsoring today's web symposium. Most of all, I'd like to thank those of you who came and spent this time with us. I hope you were able to get some information that will make life in the lab a bit easier for you. So with that, on behalf of Cambridge Health Tech Institute's Global Web Symposia Series, I'd like to wish you all a wonderful day. Bye-bye.